Hi guys, welcome back. So today we're going to be doing the DNA extraction lab. Have you ever wondered what DNA looks like? You are going to break apart the cell membrane of a strawberry and separate the DNA from the nucleus. Strawberries are a good source of DNA because they have 8 copies of each type of chromosome. This large number of chromosomes will filter out of your solution and you will get to actually see DNA. We will use an extraction buffer containing salt to break up protein chains that bind around the nucleic acids, and dishwashing detergent to dissolve the lipid or fat part of the strawberry cell wall and nuclear membrane. This extraction buffer will help provide us access to the DNA inside the cells. So for today's lab, we need a Ziploc bag with some frozen strawberries, cheesecloth. It is actually called cheesecloth because it's used in the process of making cheese. It's how we separate the curds from the whey. It's just kind of like a mesh netting. Here we go. A funnel, 2 really big beakers, 500 milliliters of water, just regular old tap water, 100 milliliters of dish soap, we're using Dawn, about 20 grams of salt, just regular old table salt, some ice cold isopropyl alcohol. This is the alcohol you might have in your first aid kit. This is not for consumption. Dixie cups, and a toothpick or stirring rod. Perfect. Okay, so this is very kitchen science. You could do this at home. It is safe, but I don't always recommend it without adult supervision. So the first thing we're going to do is place the strawberries in a Ziploc bag. We've already done that. And we are going to smash and grind up the strawberries using your fist and fingers for about 2 minutes. We kind of already gotten a head start on this, but be careful. You don't want to puncture the bag. It'll make a huge mess if it does. So now we're going to make our extraction buffer. So in one of the large beakers, you are going to add the 500 milliliters of water.
Can I see the beaker?
Mm-hmm. We are going to add the dish soap. And we are also going to add the salt. And we're going to stir this gently. We don't want to make a whole bunch of foamy mess. There we go. All right, so now we're going to add just a little bit of this extraction buffer to the bag with the strawberries, so that the soap can start to break down the lipids in the cell membrane and in the nuclear membrane, and that the salt is helping to break down those protein chains that are around the nucleic acid. So we're just going to add a little bit to our strawberry mixture. Big round of applause for Mrs. Willis today. I forgot to introduce her for those of you that might not have her in class. Okay, so we're mixing together our extraction buffer with our strawberry goop. All right, and now we are going to assemble our filtration apparatus, which we've kind of already done. We have a ring stand with the funnel, and we've placed cheesecloth inside the funnel, and we've placed another large beaker underneath the funnel. So what Mrs. Willis is going to do now is pour the strawberry mixture into the cheesecloth, and the job of the cheesecloth is to filter out all of the chunks and seeds and only allow the juice and the DNA, which is now in the juice, to pass through. So as she does this, she's gonna add more buffer solution throughout while we're filtering this to sort of wash the DNA out of and off of any of the chunks that are left over. So that's what we're gonna see here. So all of the chunks are now in the cheesecloth, and we're gonna add some more of that
Shall we
buffer.
take a look at what that looks like?
If
Or
you,
just wait?
hold on, let's see if let me move. See if I can do this without messing this up. Ooh. There we go. So you see all the chunks floating around in there, but we want to make sure we get all of the DNA possible. So we're gonna wash any of those chunky parts that we have left out with some more of that extraction buffer. Sometimes the cheesecloth might get a little
It's okay,
clogged.
it's not going too low.
So I always pick it up, woo, and let some of the, it creates a cohesion, almost like a suction. So you have to move it around. We have Mr. Stuzinski dropping in to say hi. So we might need just a little more extraction buffer. Mrs. Willis is getting all gooey.
It's in the name of science.
I love this lab. This is one of my favorite ones of the year.
Okay. I think we got quite a lot out of it, actually.
Yeah. I'm used to doing this on a much larger scale
Yeah.
for all the class. Okay, that's probably good. We can finish washing that out. And I think we've got probably all the DNA we could possibly ask for now, just in our juice jar. Our soapy, salty strawberry juice jar. Awesome. Okay. So normally, if we were in class, we would give each student a little Dixie cup, a little paper cup with some of our juice in it. Because I want you guys to be able to see everything that's going on, we're going to leave it all in the glass beaker so that we can observe everything that's going to happen. I'm going to move the webcam a little bit here, so hopefully we can see that a little bit better. Okay, so now we are going to slowly add some of that isopropyl alcohol to the strawberry mixture, and we're going to kind of run it down the side of the glass because we don't want to stir up the mixture. So because we have such a large amount of the strawberry juice here, we're going to use a fair amount of the alcohol. And then I'll see if I can get a video of what we're going to start seeing here.
All right, I think that's probably good.
See if we can. All right, so hopefully you guys can kind of see what's going on, what looks like bubbles. And then let me see if I can move this a little bit more. Okay, do we see the stringiness?
Can you just
That
put
is,
it straight down?
Not straight down, but I'm gonna try.
Okay.
There we go.
Yeah, you can really see how it's starting to form like a web almost.
Okay. Do you have your stir rod and see if you could maybe gather a little bit of that so they can see it better? Okay, do you see the strings? All right, so that's actually our DNA. We're going to see if Mrs. Willis can fish some of that out of there.
Yep, come on.
I'm holding this by hand.
Here we go.
All right, so it kind of looks like snot, but it actually feels kind of gritty. It's perfectly safe to touch, right, Mrs. Willis?
Yep.
Everything here, this is like homemade strawberry hand sanitizer, basically. But yeah, that is the DNA of the strawberry that we just pulled out. You can see there's still lots more down in the glass. And it'll actually keep separating out for quite a while. The longer you let it set, the more DNA will actually get pulled out.
Precipitate.
Precipitate, yes. All right, guys, so we'll see you next time.